great. So thank you so much to Dr. Lauri Byerley, Dr. Karyn Gallivan, and Dr. Kristin Ondrak for joining us today to talk about their ongoing project about um, dietary protein and gut microbiota, which is a mouthful for me. The microbiota one is the one that trips me up. So I'm really excited to hear the progress on this project.
All right. Well, I'm going to say that word plus another one back to back in a minute. So if I mess up, it's because Kelsey kind of jinxed me. Well, hi, everybody. Thanks for sticking around all day. Kelsey and team, this has been awesome. We've been right on time and people have been interactive. So it's really nice. And I just want to shout out to everybody who received a grant. Every one of these presentations has been totally different, which is really cool. And it's good for all of us to see all the differences, all the different types of research that can be done. So our team is back, or we never really stop, because our fearless leader, Dr. Byerley, just continues, and this is her this is her hobby in life, is to research, and Kristin and I just hang on for the ride and do what we can. But anyways, our team is back. We started this whole project a few years ago with another grant, when we were investigating protein in the gut microbiome and metabolome variations, comparing weightlifters or muscle builders, if you think about that, who either did or did not use protein supplementation. And so this grant is supporting the natural progression of that first study. So next slide, please. So here's what we currently know. Most microbes are present in our colon. And now for all of us here, if you listen to the news or read anything in the fitness world, which is in every publication online and in print, these are terms that you see a lot. We don't really know what they mean, but when you do research, we not only know what they mean, but actually what they really do, right? So we know that microbes are present in our colon. They're pretty important to us. Microbes also prefer to digest dietary fiber, and their effect on fiber has been studied extensively. So we know that. Because of that, we know that these microbes affect how we are, how healthy we are, our well-being. What we also know, switching to another nutrient, protein, we know that many Americans are consuming more protein than they need, and at the same time, not enough fiber, which is not always, well, actually never a good thing, right? Because more protein than fiber tends to reach the colon. And so we really wanted to pay more attention to protein's effect on microbes. So as a point of comparison, let's just kind of take a quick look at, so this whole slide, then let me kind of set it up a little bit. This is where we get to say, what about me, right? So the numbers on here represent a large program of surveys that were aimed at assessing the health and nutrition of children and adults in the United States. And so what we can see is on the left column, you can just see energy in kilocalories. We just tend to say calories. So that's the number of calories we take in. And then protein, fiber, and then the ratio between protein and fiber. And then going across the second column, dietary reference intake. And this is one of the numbers that you see referenced a little bit. It kind of estimates the specific nutrients used for assessing diets for healthy people, right? So in the simplest form, to be healthy, we would intake for males 56 grams of protein per day, for females 46. And I challenge most people on here to take a quick assessment of your diets and see if you're not meeting or exceeding that intake. By comparison, for fiber, you can see men and women, so 35 or 25 grams of fiber per day is a reference intake. And again, the same challenge is out there. Did we eat enough fiber on a daily basis, or do we eat enough fiber on a daily basis? And then you can see the ranges for the different ages of males and females going across to the right. And you can also see the ratio of protein to fiber. And generally it says, so males 0.3 and females 0.4, but every single one of those numbers at the bottom is more than it needs to be, right? So if you look all the way to the bottom, well, first of all, if you kind of scroll down and Lauri's kind of running the little pointer, you can see a link to the study where those specific numbers came from. So it's always interesting to see that. So if you go and take a look at some of that, it's nice because we get information on the population, and then we start to think about, oh, what could I do better, or what am I doing too much, and all of that. And at the bottom, this is where we're going next, is a ratio greater than 1 indicates that there's more protein in the colon than in fiber, right? So essentially, most of us are taking in too much protein, too little fiber, but fiber is the preferred fuel for the microbiome. So coming back around, next slide, please. So that's kind of where we're going with the study. The original paper that was published is on the right. You don't have to read that, of course, but the goal of the present study, as an extension of that, is to determine the effect of dietary protein on the gut microbiota and the metabolites that it produces. And I will let Lauri take it from here.
Yeah, so I'm going to summarize that study that we published in 2022, which actually was funded by APUS too. Thank you very much. It's great. So this data is from that paper. And again, I want to look at the protein and fiber intake. Karyn's already shown you what the typical American or Western diet contains. And so for this particular study that was, I'm going to go back here real quick on this paper here. What we did is we asked, they were all students that went to APUS or AMU, whatever, APU, and they reported that they were muscle builders, and they also reported whether they consumed protein supplements. So PS is the group that said they did consume protein supplements, and no PS is the group that did not. Now, again, this is all self-reported, and it's a cross-sectional study. And we had them just send us a poop sample and to tell us what they ate 24 hours before they sent us that poop sample. And what I'm just showing here is their diet recall information and just the limited amount of it. You can go to the paper. There's a lot more information in the paper from this table too. So you can see their calorie intake was very similar. There's no significant difference. Here's their protein intake. So we're sitting at about 118 grams of protein, and this is in 1 day or 24-hour period for the ones that reported they didn't use protein supplements. And we're sitting at about 169 grams of protein in those that said, oh, we do like to use protein supplements. And since I get to control the slides, so we decided, I'm going to go back to this real quick. You can see what the average American is consuming. And you can see here what we recommend for everybody to consume. So our muscle builders are consuming a lot of protein, which actually is to be expected because they're muscle builders. And then here's their fiber intake. So those that said, okay, we're doing it naturally, no protein supplements, they took in about 19 grams of fiber. And those that were consuming the protein supplement took in about 27 grams of fiber. So we were a little surprised that the protein supplemented group, or those that reported using them, actually consumed more fiber than those who did not use the protein supplements. But that's the way it goes. So now if we apply that same sort of principle, one thing that I want to point out from that other slide here, that it's down here in the details, about 17 to 22% of the dietary dietary protein that everyone eats every day isn't absorbed in the small intestine. So it just travels on through and it reaches the colon. And so then the bacteria in the colon, that's what we study, are primarily bacteria, but all those microbes that are present in the colon, they've got protein to do something with. And you can see for our study here, if we assume about 17% of the protein was not absorbed in the small intestine, we could have done 20%, 22%, but I think it doesn't really change the numbers that much. So for this no protein supplement group, about 20 grams of protein reached the colon. And you can see they only consume 19 grams of fiber. So they've got more protein present in the colon than they do fiber in the colon. So that's where we really got interested in what impact does that have? And so we collected our poop samples, and this is metabolites that are in the feces. So there were about 170 metabolites that were different. And the ones that were most different were allotonin. And you can see, if you look here, we've got our little chart here, the red is really high, and it's really high in this protein supplemented group. And so they had more protein, and they had a little bit more fiber, but they're going to have a lot more protein compared to fiber. And so I think there's about 12 or 13 on this list. These are the very top of that whole list. You can also see uric acid is here. There's a few of the other ones there that I'm not going to talk about at all, because what I really want to focus on is this allotonin and the uric acid. But I will point out, for those of you who consume non-caloric sweeteners, there's sucralose. We picked it up in the feces. But anyway, that's not what we're studying or looking at all. So what is allotonin and what are uric acid? I'm sure you all have heard of uric acid, and maybe you've heard about it in relationship to gout, because that's going to be one of the things that the doctor is going to tell you. You have gout, you got too much uric acid on board, you need to reduce the purine in your diet. And so what are purines? There's 4 of them that you'll find in your diet, and that's adenine, guanine, xanthine, and hypoxanthine, of which I don't expect any of you to remember, but you all know DNA and RNA, and purines are the backbone, or one of the backbone molecules for making RNA and DNA. So what foods will you find these in? Any living creature, plants, animals that you eat will have RNA and DNA in it. So things like milk, for example. Milk, it doesn't have purines in it because it doesn't have RNA or DNA in it. But as we show here in this picture, we've got meat and we've got peas. So those will have purines in it. Both protein and purines are unique in our diet because they contain nitrogen. We won't talk about that at all today, but we are going to be looking at that as well. But what I want to focus on here is purines. So you eat the food, and that goes through your stomach, and it goes into your small intestine. And that food is going to be broken down into, for proteins, that'll be amino acids, purines, it'll be for purines will be broken down from the DNA and the RNA. You've got carbohydrates and fats and everything that are going to be in there that are all going to be broken down, but we're not focusing on carbohydrates or fats. So the purines actually can be absorbed from the small intestine along with amino acids. But again, I want to focus on purines. Those absorbed purines, they can be used because you might want to build some DNA and especially RNA. You want to be building RNA molecules from DNA. And when you don't need it, then it's broken down to uric acid. So this is where you're going to get a buildup of uric acid, and it can cause gout in those who don't get rid of the uric acid appropriately. So purines tend to be higher in animal products than they do plant products, although there are a few plants that are pretty good sources of purines. And so that doctor who's telling you, okay, I need you to reduce your uric acid intake so we can lessen those gout attacks, they're going to focus on an animal protein-rich diet. Okay, so that's the uric acid. Now, uric acid wasn't at the top of this previous figure that I showed you. It was allotonin. Well, allotonin is a breakdown product of uric acid. We don't have the enzyme in our body to break uric acid down to allotonin. So it's uricase. It's the name of that enzyme. What we do have, though, are ROS, which are reactive oxygen species, which I'm sure you've all heard of too. That's been a big thing. So that's where we're pushing, you got to eat your antioxidants. So have your plant foods that are rich in antioxidants. So that'll bind up these ROS. There are actually a lot of them electrons that are just hanging around and bind those up, and they're not available. So then you're not going to be converting uric acid to allotonin. Right now, they are looking at allotonin as a marker of inflammation. We'll see if it works. So it would be like C-reactive protein, which you may have heard of that. So this allotonin is very water soluble. Uric acid is too, but not quite as good, and it tends to crystallize. So that's where we get into gout. But allotonin then would be excreted in the urine Or its other fate is it can be dumped into the colon then. So we've got this big colon here. So what's not absorbed in the small intestine is going to enter the colon. So for example, the protein that we talked about will enter the choline. These purines will enter the choline as well. And so in the choline, then the bacteria, they have all these enzymes, so they can make allotonin. So that's one of the things that we're looking at with this current study is if we give more protein, again, I already just told you that protein sources that are rich in protein are going to have more purines as well. So we're hoping the purines are just going to come along so we can look at them. And if we give people more protein, then as Karyn indicated in our, when she gave you the goal of the study, what factor impact does that have on the gut microbiota or the metabolites that it produces? So we designed this randomized clinical trial. And first what we do is recruit potential participants. And Dr. Ondrak will talk about that in just a minute and give you some details on it. From those participants, then we screened them. The ones that were eligible then were randomized to one of two groups. And what we did is we asked the participants to consume 50 grams more protein a day. Now, it's obviously difficult to tell people, okay, I want you to eat 50 grams more chicken a day, because then we'd have to ask them to go out and buy chicken. So we decided that what we would do is we'd use a protein supplement and provide that to the participants. It's a little bit cleaner, and we're providing it to them, so it's free to them. So we decided to look at two different ones. So we looked at whey protein supplement, because again, like I already indicated, dairy doesn't have much purine, but it's got lots. This will have lots of protein in it. It's all protein. And so then we don't have a supplement with purines. And then the other one is a pea protein supplement. And peas, as you saw on the previous slide, you saw a little picture of peas. Peas do have a lot of purine in them too. So we're hoping that we're getting purine and protein here with this group. We had them consume this for 7 days. We thought that was probably going to be long enough. Again, remember, they're adding it on top of what they already ate. And then Kristin will tell you what some of the other methods that we used here to assess them. So I'm going to turn this over to Kristin now, who's going to tell you more about the study. I saw Dr. Ondrak here. Oh, maybe
she is here, but we can't hear her.
Huh?
Can you hear me now?
Yes.
Yes, there we go.
Okay.
Gotcha.
I was rambling. All right. Sorry about that. All right. Everybody can still hear me? All right. Sorry about that. So this slide shows our recruitment efforts. So the exclusion criteria are listed in table 1. So specifically, if they were less than 25 years old or taking any pre or probiotics in the week prior to beginning the study, they were not able to participate. If they were taking any prescription medications other than oral contraceptives, if they had taken antibiotics in the last 3 months, diarrhea inhibitors in those last couple of weeks, laxatives within the last week, any other dietary supplements, because of course we don't want any sort of added variables or variants in there changing the gut microbiome. If they had been diagnosed with cancer, which of course, even if the tumor wasn't located in the gut, because that can certainly change the gut microbiome. And if they had any sort of inflammatory bowel disease or disease of the gastrointestinal tract. So on the right side of the slide, you'll see our consort diagram where we sent over 9,000 emails to undergrad and graduate students at APUS. We spread personal communications, so personal emails to friends and word of mouth, and we estimated reaching 60 people that way. And then reaching an additional almost 500 through social media, LinkedIn and Facebook posts. And then we estimated 2,339 views of those posted advertisements within the university and outside community. So we really tried to spread this word. And when you see how many participants we ended up with, you'll see it takes a lot of outreach to get folks that are going to be compliant with a longitudinal study, as we needed them more than one data point. Next step was enrollment and randomization. So of all those folks that either saw or read our messages or recruitment efforts, 225 completed the survey and were screened. You can see 138 of those were excluded. The first group there because they didn't fully complete the consent form, and then the majority there didn't meet the inclusion criteria. So that was more common. Of those that moved on, we're following the green arrows now, 87 were eligible, met the criteria, and were successfully randomized to either that whey or pea protein group. 20 were excluded from that because of just not responding even after the randomization. And frustratingly, some were even sent all the wonderful supplies and then kind of fell off from communication. But that happens, as we know, with human subjects. 67 responded to our communication. One fell off there, being able to not receive, or we couldn't ship them the supplies. So 66 were shipped supplies, and you can see the 4 that were excluded from there. It either didn't respond after receiving supplies, did not collect the fecal samples on the right day, or they decided they didn't want to do it after seeing what the fecal sample collection involved. Or decided they couldn't collect that sample or provide that sample when needed, which we'll get into that in a bit here. So from that, we were down to 62 active participants. 4 were excluded, 1 due to the fecal sample just not being usable, too green and viscous, 3 due to shipment delays. I know I'm grossing everybody out this afternoon, right? And then one of them went camping for 2 days before the final collection. So as you'll see in a moment, the timing of the first fecal sample and then the 7-day supplement period, it was very crucial that they followed it very rigidly and then provided that last sample. So some folks weren't able to do that. But we did successfully get these 62 active participants. You can see if we follow it down here, there were 28 successfully completing in the whey protein group and 31 in the pea protein group. Okay. You want to go on to the next slide, Lauri? We could break down more details there, but for the sake of time, I better keep moving. So here were our data collection methods, which Lauri showed on the nice timeline. But in terms of collecting what food they ate, so we did a 24-hour dietary recall using the ASA 24, which was completed online, nice validated standardized instrument. We used the International Physical Activity Questionnaire to get an idea of their physical activity information. Of course, they weren't changing anything. We were just trying to assess what is their normal pattern and amount of physical activity. We did some survey questions on Qualtrics just to get demographic information. And then to collect the fecal samples, we used this biocollective system. So you can see it came with rubber gloves, a small cooler of sorts that they received and then shipped back to us, a hammock made of paper that you put over the toilet to collect the sample into, and then, of course, biohazard bags. So they went into the hammock, and then the whole hammock was put into the plastic bag and shipped away. They didn't have to deal with any of that. Dr. Byerley is graciously doing that at this phase of data collection. But in terms of the participant burden, I do think it was pretty straightforward. And from our previous study, we even made a video demonstrating how to do some of that collection. In terms of protein supplement, you can see now Foods generously donated pea protein and whey protein. And if I'm recalling, each participant got this big size bottle of unflavored chocolate and vanilla. So we couldn't we gave them recipes and ideas and all, like, lots of freedom with how to consume it, and lots of suggestions in case taste or flavor was cited as a barrier. We tried to nip that in the bud. Next slide, please. So what we're doing now, the work in progress, the chemical analyses, and then that bioinformatic and stats, or stats once we get some of the data. In terms of the chemical analysis, Dr. Byerley is running the charge here with this, with her colleagues at LSU. So doing a 16S RNA analysis to look at fecal bacteria, metabolomic analysis to look at the metabolites present in the fecal matter, fecal nitrogen analysis to determine protein content, and then lastly, the purine analysis of the dietary supplement. So lots of moving parts there, lots of different analyses and different stages. In terms of the bioinformatic data, we're trying to determine if increasing that dietary protein intake by 50 grams a day increases the fecal allotonin content as we expect. And then our other big goal is to identify if there is a bacteria and metabolite pattern that's consistent with eating dietary protein. So the two big goals. Next slide. Our preliminary results of our randomized participants. So got this great graphic on the left showing from where our participants were derived. So you can see California, reminds me of the electoral map now that we look at it. California, Florida, North Carolina had our the most participants, followed by Virginia, New York. And the different shades are to indicate where the participants are coming from. So as you can see, we reached a lot of different states. And we're pretty proud of our recruitment efforts and being able to pull this off completely remote and with no home base. You can see, and on average, the whey and the pea protein group had similar ages, 36 versus 38 years, 20 males and 17 females in the whey, 15 males and 24 females in the pea. You can see the ethnicity breakdown in both groups. The majority of participants did not identify as Hispanic or Latino. And then you can see the racial breakdown. The vast majority of both participant groups being white. The second most numerous would be Black or African American, followed by Native Hawaiian, Pacific Islander, Asian, American Indian, or Alaska Native. The height, we did not do that on purpose, but they have the exact same height between the two groups, or same mean height. And you can see the weight similar between the groups. BMI slightly higher in the whey versus pea. We certainly did not randomize based on any of those data. Next slide, please. So our conclusions at this point in the study, we were successfully able to recruit participants virtually through word of mouth and lots of emails and trying to get the word out. We demonstrated that a randomized clinical trial can be done successfully in a remote setting. I do feel like the number of these types of studies are growing, but it's challenging. And we exceeded our statistically determined sample size per group. So for that whey group and for that pea group, that was our finish line, getting beyond those, what our power analysis showed us in terms of what we would need in each group to show statistical significance if it exists. Next slide, please. So we want to say a big thank you. We're honored to receive a 2024 APUS Faculty Research Grant. And we also want to give a shout out to Now Foods, who generously supplied that protein supplement and a whole lot of it to all those different participants.
We should also say we got a 2023 and a 2022 grant for this study. This isn't the kind of study you do just in 6 months.
Absolutely. Yeah. So I don't know how we're doing on time. If there are any questions, our emails are there if there's not time.
We got 2 minutes. If anybody has any burning questions. Maybe 3. I really appreciate you talking about sort of the logistical challenges of an endeavor like this. It's not something that you often think about. And it's such an ask, right? I mean, you make it as easy as possible, but still it's quite an ask to have somebody, add something to their diet to consume that much protein, and then the other side of that.
And we also we learned from our first study, and we really tried to mitigate product loss. You know, it's a kit-based study, if you will, and you know, we lost more the first time than we did this time, which was really nice. But that really comes down to communication, and each subject was assigned a handler. So we were in contact with that person from day one, you know, first poop sample, and then every single day that they were to consume protein, the reminders were sent, and we helped to troubleshoot as well until it was over, and they like put it back in FedEx, and we were able to track it back to us. So we really we did better this time. So
it also is really interesting. So I had some colleagues, not Kristin or Karyn, that kind of looked at it and said, well, what are your exclusion criteria? We have very strict exclusion criteria. And today, in today's world, trying to find people who aren't taking prescription drugs is not easy. And but we managed, we did it. And you'll notice our age was 38. So 25 was an exclusion criteria. So we weren't looking at the younger people who would probably not be taking drugs, although I don't know if that's an accurate statement anymore either. Yeah. Another challenge too that none of us expected, FedEx. FedEx is about 70% good at doing what they ought to be doing. I chased around New Orleans for one sample. And finally,
there is a question in the chat, Lauri. What is the attrition rate with the diet? And this is from Tim White. Tim, do you mean like the attrition rate with people who were taking the supplement itself? Yeah. That's a good question.
So that was pretty low. So I would account the attrition rate would be those who decided they couldn't do the supplement. And Kristin and Karyn can answer that question a little bit better because they did all the follow-up with the participants. But usually we lost them at the very beginning if we lost them, and that wasn't very many.
No, it was low. It was pretty low, which is really nice. Well, Tim, that might be the next study. How many have continued to use
use the protein supplement, do you mean, Tim?
Yeah, that was the question. How many have continued to use?
Yeah.
Well, I would assume that's the protein supplement, which actually that's not so much what we're interested in. We'll have to.
Yeah. And we didn't have that question, so we don't know. But because some of the people we recruited were friends and family, I do know that some people have continued to use just protein in general, because for a variety of reasons. And then others have switched their standard brand to Now Foods because the whey protein itself was such a pure, easily mixed supplement, which is kind of nice to hear. So
yeah,
the win for Now Foods.
Yeah. And this wasn't an advertisement for Now Foods, so.
No, not at all.
Yeah.
Look at that, Jackie.
Oh, that's great, Jackie. Yay.
So
awesome. Well, thank you all so much. This is more out of my wheelhouse as far as subject goes, but
what about the fiber?
oh boy.
So we would in future studies we would continue to look at the protein, but what we need to do is we need to shift so that there's more fiber and less protein, and we could connect the protein to maybe other kinds of chronic diseases would be one thing, or we're also interested in body composition. So what role does this play in body composition? So we're thinking about and talking about what our next role study would be, but we got to have the data from this one first. Hopefully that answers your question, Richard.
Great. Thank you all so very much. And we have our last team queuing up to finish us out today.